So here we talk about specifically the type of gene mutations. Now the type of gene mutations in which the smallest level of mutation you can think of is called point mutation. It happens at a point. It is a very minor change in the sequence of the DNA, in the sequence of the gene that is involved. It may be such that one of the nucleotides or one of the nitrogen bases get replaced by the others. That is referred to as base substitution. Base substitution are of two types, that is transition and transversion. If a purine gets replaced or substituted by a purine, then it is called transition. However, if a purine gets substituted by a pyrimidine at a point on a gene, then it is referred to as transversion. So remember that what nitrogen base has to actually be there in a gene, that is replaced or substituted by another nitrogen base, which would alter the message on the DNA. And you all know that the message on the DNA in the form of a genetic code is copied onto the mRNA. And then there can be frame shifts. In frame shifts, there is complete loss of a nucleotide. Say for example, there is a deletion of a nucleotide in the gene, or there can be an addition of an extra nucleotide, which is referred to as insertion. So deletion and insertion will again alter the message that is present on the DNA strand, which is required for protein synthesis, and such deletions and insertions lead to a change in the entire reading frame. The reading frame refers to the message that ultimately gets copied onto the mRNA and which is read by the tRNA during translation. So there is a shift in the reading frame, either because there was an addition or an insertion of a nucleotide on the DNA strand, or there was a deletion or removal of a nucleotide on the DNA strand.